Good morning, everyone. Good morning. Welcome to this particular lecture series. Today we are focusing on this particular aspect of immunology, and today we are talking regarding your MSC molecule. So, see why we are talking about MSC molecule. In past many lectures we have discussed regarding this molecule, whether in terms of your antigen presentation cells, whether we are talking about macrophages, B cells, dendritic cells, or when we are talking about exogenous and endogenous antigen, whether it's the difference between the exogenous and the endogenous part that we are talked about. So today we'll be taking a journey where first of all we'll be try to understand that how this particular MSC molecule was discovered, what are the different classes of MSC, what are the structural features of this particular MSC molecule, and how exactly the antigens are being presented by this particular molecule on the surface. That is in nutshell what we're going to learn in this particular lecture. So let's start with the brief introduction. See, what is MSC molecule? I'm talking about MHC. So full form is major histocompatibility complex. Now, C is the most crucial alphabet in this particular aspect because we are talking about a complex. We are not talking about an individual peptide. So if it's a complex of multiple peptides, it has to be coded on by certain genes. And those genes, each gene is coding for a single peptide. So there are multiple genes. So what we are talking about, gene clusters, gene clusters present on the chromosome, which are producing the products or the proteins, which are actually very crucial in performing its function in immunology. And the major function what it performs is to differentiate between self and non-self. How immune system, how your immune system recognizes which is foreign, which is my own, that sensor point, that defining molecule, that molecule which puts an authority that this is outside and this is from inside, that particular molecule is MHC molecule. This particular complex determines whether it's a self antigen that you should not put on, or it's a non-self antigen that we should talk about. So this is the scenario that we'll be discussing in this particular aspect and in this particular lecture. So this MSC is again, as I told you, is made up of multiple loci. In fact, you will see in the course of lecture that is present on the chromosome number 6 in case of humans, and in that region also it contains multiple genes. More than 200 genes are present in that particular section, which is giving rise to different classes of MSC molecule. So that is exactly what we are trying to accomplish in this particular lecture and in the subsequent lectures to come. So why we call it as histocompatibility? Because initially when it was discovered on the basis of graft rejection. Now what is graft rejection? Let me take you to this particular slide. I'm talking about transplanting certain graft. Now suppose there is a donor, this particular recipient which will be receiving, suppose I want to graft the portion of a skin on these animals. So in order to accept the other individuals and component or tissue, the recipient immune system should say yes, then and then only this will be accepted, otherwise it will not be accepted. Who decides what to take inside our body and what not to take inside our body is our immune system in terms of your transplant or graft transplant, what we are talking about. So what they did, how exactly the experiment went ahead. The donor suppose has this particular genetic combination. Then they took a recipient which is exactly identical copy of the genetic combination, and the other recipient which is non-identical. There is a certain difference. They observe that the graft is accepted by the immune system in case of the genetically similar, identical, I should not say similar also, genetically identical recipient, whereas the graft is rejected when there is a disparity or dissimilarity between the genetic combination the immune system recognizes that particular skin as foreign or non-self or outside, and it is considered as an antigen against which the immune reactions or immunological reactions are mounted. So that switch, that decision is provided by your MHC molecule, and that is the reason why we call them as histocompatibility. Histo means tissue, compatibility means whether we are compatible to attain or to accept that particular tissue that is being given, and that is why the name came as major histocompatibility complex, because it is either in accepted or rejected form that we are talking about in this particular section. Then let's have a brief history what's been done in the past. So originally it identified as, people originally recognized it as an antigen molecule that is present on the blood cells in 1930s. Then they further showed, they further characterized this particular molecule on the basis of graft reaction, the experiment which I am telling you right now, performed by Snell, and he was awarded Nobel Prize for this particular experiment in 1980. Then multiple works have been done in this particular category, taking out the MHC, studying its genetic part, studying the complete structure of the MHC molecules of all the classes, identifying individual classes. So there, the work has been going on continuously in this particular series. But let us see historically what are the milestones that we say in terms of your MHC discovery or MHC expression of its system. Then the work done by the Rolf and Doherty in 1970, what they reveal? They reveal that this MHC molecule, along with the antigen, is exactly the complex which is recognized by T cells in order to elicit the immune response. So first of all, Rolf and Peter, these two were the scientists who exactly told us that yes, MSC is involved in presentation, and the MSC molecule conjugated with the peptide is actually the molecule that is recognized by T cells in order to elicit the immune response. And for this work, again, they were awarded with the Nobel Prize in 1996. They clearly identified that yes, there are two classes of MSC molecule, class 1 and class 2, whereas these days we are aware that there are three classes of MSC molecule, 1, 2, and 3, and we'll be studying all those three. So the two classes, classically what they identified was to be contained with different chains. Class 1 contains two peptide chains, alpha and beta 2. We'll be seeing this is just a glimpse that what they discovered, and then class 2 contains alpha and beta chain both. Then this particular aspect is done, that how you will be defining this particular histocompatibility complex. See, it's a group of genes. It's a group of genes that is coding for the peptides. Those peptides will be expressed on the cell surface, and those peptide molecules forms a complex called as major histocompatibility complex, and their main work is to determine the tissue type and the transplant compatibility. Now, when the definition is being done, what we understood? What we understood is that there is a cell, on its surface certain molecule is there. This molecule is actually responsible for determining self and non-self, and it is also responsible in turn to accept or reject the graft that we are trying to put, and that molecule is called as MHC molecule. Now the question comes, what type of cells am I talking about? See, MHC molecule is present in all cell types. That is the reason why it is acting as a switch in order to determine, because it is present in all the nucleated cells. I have already discussed this particular aspect in previous lectures. See, it has two classes that we are talking broadly, MSC 1, MSC 2. We'll be talking about third in the later section. Now, this particular 2 is present in all the APCs, antigen presenting cells. Whereas when we are talking about your other aspect, when we are talking about your other aspect, it is present in different scenarios. So yeah, we are back online. I think there was some power glitch. So that is the thing that we were discussing, that how this particular section is being raised. So yeah, so we when we had established this particular aspect, so the second MSC molecule is present only in antigen presenting cells, that is macrophages, B cell, and DCs that we are talking about. Whereas MHC1 is present on all nucleated cells. Why I am writing nucleated cell? Because it is absent in RBCs. It is not present in erythrocytes. So that is the thing that you should clearly remember. M is major, H is histocompatibility, C is for complex. And the reason why we gave it the name, because it is expressed on the surface. Now please understand, when I am studying immunology, I am an immunologist, then this particular name MHC is just a common generic name that I'm using. Whereas in technical terms, we call it as an antigen molecule because it is expressed on the surface. So I can use it as an antigen molecule to determine the marker. To determine the marker. So in humans, it is called as HLA complex, that is human leukocyte antigen. For example, if we talk about in the swine, we call it as SLA, swine leukocyte antigen. In mouse, it is called as historical name, which is provided is H2, that is histocompatibility 2 complex. So these nomenclatures are very crucial in terms of your competitive exams. Multiple times the question comes that what is the region, what is the common name, what is the general name of the complex that we are talking about MSC in human, that is human leukocyte antigen. In mouse, it is called as H2, that is histocompatibility 2 complex. So this is the thing that you should be able to understand clearly, that these molecules that we are talking about, the peptides that are expressed on the surface of this particular part, this is how the peptide looks actually in the real-time scenario. When I'll be showing you in the picture, this is alpha chain and this is the beta chain. So this particular molecule is protein, but it's a glycoprotein. It's a glycosylated protein. A carbohydrate moiety is attached to the protein. So the post-translational modification that is happening after the genetics, after the translation part, is glycosylation, and then it is expressed on the surface of the cell. And based on this particular arrangement of these chains, we call them as class 1, or we call them as, or we calling them as class 2. So it's a very large, if you're talking about the genetic part, it is present on the chromosome number 6. It contains the large genomic area divided into 3 specific regions in case of vertebrates that we are talking about, and in where we'll be discussing about the mouse genome in detail. Then broadly, currently we are aware that yes, we have 3 classes of MSC. We have always talked about 2 classes, but when we are talking about the genes, in terms of genes, we'll be talking about 3 classes of genes, that is class 1, 2, and 3. Class 1 is a glycoprotein. It is coding for a glycoprotein which is expressed in all the nucleated cells. Its major function again is to process the antigen and present it to the cytotoxic T cell for cell mediated immunity. Class 2 is again a glycoprotein present in macrophage, B cells, and dendritic cell, and its main function is to activate the helper T cells. What is class 3? What is class 3? Now see, class 3 genes produce proteins. These are secretory proteins. That is the reason why we are not talking, why we had not talked about these proteins in terms of the surface expression. On the surface, the work of presenting the antigen, that is the job of first and second class. Third class of MSC gene is secretory gene. It is secreted out of the cell. It is secreted out of the cell. And secretory functions, what do you mean by secretory function? These proteins, once they are secreted out, they will be performing different functions in complement pathway, in inflammatory reactions. Yes, they are mediating the effect of immune system. So that is the reason why we are not talking about this particular class till now. Fine. So what is the basis that we're talking about of the MSC system? The basis is very simple. It is based on the genetic characteristic, like in men or in humans or in mouse that we're talking about most of the species. Each class of MSC is represented by different genes. Like if I'm talking about class 1, it is made up of 100,000 kilobase pair of region or 200 kilobase pairs of region containing multiple genes. multiple genes, and that is the reason why we are calling it as human leukocyte antigen. The class 1 contains, if you bifurcate the scenario, then you will see that this is the representation or chronology. You will see we have a chromosome number 6, and it is present on the short arm, that is called as P arm. On the chromosome 6 P arm, we have three defining regions that define this particular MHC gene, and those regions are 1, 2, and 3. And we'll be studying those 1, 2, and 3 regions. 1, 2, and 3 regions. The size is represented here. That class 1 is quoted by almost 4,000 kilobase pairs of region, whereas the class 2, what we're talking about, is exactly, if you talk about, is 1500 kilobase pair of region, the class 3, and the class 2 is around 500 kilobase per region. You can see the number of genes that are there. That is the reason why we call it as polygenic. So for what do you mean polygenic? Multiple genes are coming together to give rise to a single functional MSC1 or MSC2 that we're talking about. Multiple genes are coming to give rise to MSC1, and similar is the case with MSC2. So in humans, it is present on the chromosome number 6, whereas in mice, it is present on the chromosome number 17. And that is the thing that you should have a very clear understanding, that the chromosome number, what we are talking about, is 6 and 17, present on short arm, present on short arm. So if you see here clearly that the region what we are talking about, the longest region is of 2000 kilobase pairs, and the other two regions, the second and third, are very short regions that we are talking about. The class 1 MSC molecules are recognized by different regions. Now let's see class 1. I am talking about this particular class currently. The class 1 region is determined by A, B, and C region. So E also present, whereas class 2 is recognized by DP, DM, DQ, and DR. That is a representation that you should always remember, that if there is a symbolic nature of DP, DM, DQ, and DR, that means the immunology, in immunology you are talking about class 2. Whereas if you are talking about class 1, then it will be represented in form of HLA, human leukocyte antigen A, B, C, or E. That is the thing that we'll be talking about, and that is represented in this format, that we always represent ABC for region 1, and we represent DP, DQ, DR in terms of region 2. Now, what is this DP, DQ, DR? See, these DP, DQ, DR are the nomenclature for the regions that are present on the chromosome number 6, representing the MHC molecule. So these are the regions in which different genes are present, and these genes will be coding for a functional MHC molecule. So this is what? This is a map. This is a genetic map for MHC loci, and we call it as MHC loci genetic map, human MHC genetic map actually. And there is multiple molecules, there are multiple polymorphism that will be seen throughout MHC molecule, because it is there are single nucleotide polymorphism. See, polymorphism will be there. Why? Because there are multiple genes that are involved. So any substitution reactions that are happening in between the genes will be giving rise to single nucleotide polymorphism, and that is the reason why we say that MHCs are highly polymorphic, and that is the reason why, because of this polymorphism, individual to individual acceptance of the graph is varied, and that is, this polymorphism arises because of these reasons, because it contains multiple gene loci, and among those genes there are multiple alleles that are present on different loci. And the another genetically important aspect of MHC is the codominance. Perhaps that is the most unique aspect of MHC molecule, that is codominant. What do you mean by codominant? Both the alleles are expressed together. Both the alleles are expressed together. So if there is a variation in any of the allele, you will see the polymorphism is immediately expressed because of the codominance. So what do you mean? How alleles are being constituted? So if we talk about class 1, we have ABC, HLA-A, HLA-B, HLA-C. That's what discuss the name of the regions on the chromosome. Now this particular region contains almost 218 alleles. 218 alleles. And this is the tally that I want to show you, just to show you that how many alleles are there. And these alleles are expressing codominantly. There is no case of heterozygous nature A and A. No, there is a codominant nature that we are talking about, and that is the reason why we see multiple polymorphism. Like if you talk about your class 2, then we are talking about DP, A, DQ, and DR regions. These are the three regions that we always talk about in if we are talking about human leukocyte antigen. You see, cumulatively it contains almost 100 alleles. Cumulatively it contains almost 59 alleles, and the number is again increasing in the third region. So higher the number of alleles, higher is the polymorphism that will be seen, and that is the reason why MHC molecule is highly polymorphic. And genetically, we always determine that as a very high hotspot of polymorphism, hotspot of polymorphism. Now, this is how you see RBCs, no MSC, APCs, antigen presenting cells. Now, please remember, now majority of the times students get confused in this aspect. What kind of MSC molecules are present on APCs? What kind of MSC molecules are present on APCs? 95% of the students will be saying MSC class 2. But please understand the question. The setter is asking you a simple question. What kind of MSC molecules are present on antigen presenting cells? Now, what are antigen presenting cells? B cells, macrophages, and dendritic cells. All these cells have nucleus also. And I told you, nucleated cells, all the nucleated cells will have MSC class 1. So the APCs contain both MSC1 and MSC2, and this we have discussed when we were talking about the cell-mediated immune responses, that these APCs can elicit the humoral response also, and these B cells specifically, when we are talking about B cells, they can elicit the humoral response also, and can elicit the cell-mediated response also, trigger the cell-mediated response also. So please remember, APCs contain both the types of MSC molecules, and that is the reason why we are calling it as that. MSC1 is a glycoprotein present on all the nucleated cells. MSC2 is present on the APCs, which already contains MSC class 1 molecule, and that you should always remember. And the cell types that are devoid of MSC is RBCs. It's RBC. Fine. Then what about class 3? Now, class 3 are, I told you that it's a secreted proteins. It performs various functions like multiple cytokines that we are talking about, TNF alpha, complement proteins that we'll be talking about in subsequent lectures. Then this expression, when we are talking about 1, 2, and 3, the expression of these MSC molecules from the gene, this is increased by the cytokines when we are talking about any kind of eliciting any kind of immune responses. Fine. And then if you have a gene, suppose if you have a gene, you have this particular MSC molecule region here that we are talking HLA region, then its promoter segment will also be present here. Now, some factors are required in order to allow the binding of RNA polymerase so that this particular region can be expressed. That those factors are called as transcription factors, and in case of MSC, the trans activators are RFX and CTA. So that is all about the brief introduction regarding MSC molecule. And let us now talk about specifically structure part of class 1 MSC molecule. Class 1. So you see, this is how the class 1 molecule is represented. There is one chain and the second chain is here. The first chain is called as alpha chain, which contains three domains that is alpha 1, 2, and 3. Now this is a glycoprotein chain. This is also glycoprotein chain. This is also glycoprotein chain. The characteristic feature of MSC molecule is this loop. These loops are belonging to immunoglobulin superfamily, and that is a reason because of the presence of these domains, we categorize them under the immunoglobulin superfamily. Now, this particular beta 2 microglobulin is of 12 kilodalton, 12 kilodalton, and when we are talking about this alpha chain, it is of around 44 to 47 kilodalton uh, protein chain. Now, the question comes again: what experimental setting will you do to identify or to prove this statement that beta chain is 12 kilodalton, alpha chain is 44 kilodalton? Very simple. It's very simple. You take any cell will be having MSC class 1. You take out the membrane fraction. From the membrane fraction, you take out the protein content, or you take out the MSC molecule. Once you have taken out the MSC molecule, you run it on SDS page, you will be getting only two bands. Two bands, one band will be of 45, 46 kilodalton, and another band will be of 12 kilodalton. So you will be seeing, you will be clearly saying that yes, the MSC class 1 molecule is made up of two separate peptide chains. One is 12 kilodalton, that is light chain, and another one is 45 kilodalton, that is heavy chain. And this is how the discovery or the structural aspect of this particular MSC molecule was determined. Now, another unique part that you should remember with this pictorial representation is this region. You see there is a clear cut cavity or the cleft that is there for antigen binding, and this is closed cleft. This is a closed cleft where the processed antigen will be binded and will be presented on the surface. Now, this alpha chain is made up of three regions, 1, 2, and 3, and we call them as ABCs. The cytoplasmic domain is only because of the alpha chain. There is no cytoplasmic domain of microglobulin beta 2 chain, and this particular cytoplasmic domain contains almost around 90 amino acids. So it's a 90 amino acid long tail that we're talking about. The transmembrane region that is present in this particular section is roughly around 25 amino acids. 25 amino acids. So that is the thing that you should clearly understand the portion, how this particular structure is being varied. Now, how this folding is done? The folding is done because here there is a cysteine residue, here there is a cysteine residue, and this is nothing but a disulfide bond that we are talking about. This is a disulfide bond which has resulted in the looping of it. So the MSC molecule, MSC class 1 molecule contains 3 disulfide bonds. Always remember that it contains 3 disulfide bonds. One is present in beta 2 and 2 are present in the alpha chains. So if you look at the complete structure representation, the complete structure is deciphered, and it looks like in real time in this particular format. And this particular cavity is called as peptide binding cleft or peptide binding region, and that is how we represent in a planar form the complete MSC molecule. You can see here that beta 2 microglobulin does not contain any transmembrane or cytoplasmic domain, whereas alpha chain contains this particular region, which is 90 amino acid, 25 amino acid of transmembrane, and 30 amino acids of cytoplasmic tail. And that is why we call it as a heterodimer. Why? Because it is made up of 2 chains, alpha and beta. That is the reason why we are calling it as a heterodimer. So always remember this particular aspect, that how it is. Then another important question, if you see this particular image, let me erase the complete representations here. If you see this, now the question comes, if this is a complex, how it is structurally stable? We know that peptides can diffuse, but there is no attachment between these 2 chains. Then how they are in the immediate vicinity to form a complex? What is the interaction that is there which is holding this particular MSC2 molecule? Then those interactions are non-covalent interactions. Always remember that the beta 2 chain is attached non-covalently with the alpha 3 region of the MSC class 1 molecule, Fine. Now see, this particular beta 2 microglobulin actually is coded by a gene that is present on chromosome 15, whereas these 3 genes are the main MSC class region which were present on chromosome 6. So these are the products of two different segments that are come together to give rise to a class 1 molecule complex. So always remember this particular part. Always remember this particular part. And then another important aspect, this particular alpha 3 is the region which will bind to the receptor on the T cell surface and the receptor of CD8. MSC class 1 binds to cytotoxic T cell co-receptor, that is CD8. Now CD8 receptor will always be specific for this particular alpha 3 region. So that is that thing you should clearly remember. And we will not, I'll not push you again to MSc class 2. First of all, your job is to understand clearly the MSc class 1 so that we can step ahead onto MSc class 2 and remaining genetic diversity of MSc. So your job for today is to read the complete lecture, go through it once, and then try to inculcate what we had discussed in this particular setting, in this particular lecture, so that we can again mount on this lecture and can go ahead to describe the different aspect of this particular series. So these MCQs will be helping you in order to better understand the topic and in order to summarize what you have read till now. So the MCQs are always a very good tool to understand a topic. So thank you so much for patience listening. We'll again meet in the same set of series with the narration and the completion of the aspect of the MSc molecule. Till then, take care and wish you very happy learning. Thank you so much.